So one of the unique aspects of our research is that we have privileged access to every tissue that HIV might encounter during sexual transmission. So as this slide shows, we have all of the tissues of the female genital tract, the male genital tract, and the anorectal tract. And the great thing, particularly if you look at this anorectal tract picture, is that we are able to compare different mucosas from the same donor. So here you can see the type 1 mucosa of the rectum versus the type 2 mucosa of the anal canal. And we have fully optimized mononuclear phagocyte isolation protocols from these tissues, and we published this in the Journal of Leukocyte Biology a couple of years ago. And to cut a very long story short from this paper, we basically compared a whole range of enzymes and looked at the effect that these different enzymes had on cleaving surface receptors. And what we found was that trypsin, which is what is commonly used to digest these tissues, especially the epidermis, actually cleaves many mononuclear phagocyte markers, especially CD1C, CD11C, CD14, and CD141, as well as the HIV entry receptor CD4. And if I can direct your attention to the right-hand side of the graph, to the flow cytometry section, you can see here that when we digested epidermis with trypsin, you can see that you don't see any CD11C positive cells. If you digest it with the more gentle collagenase type 4, a CD11C positive cell population appears. And we were able to use these strategies to show that the epidermis doesn't, as previously thought, only contain Langerhans cells, which are here shown in green, but also contains two other mononuclear phagocyte subsets. And today I'm going to be talking to you about the CD11C positive ones, which are shown in blue. And you can see using a whole range of markers on this flow cytometry plot, we can identify them, but especially CD11C, which we find is the best way to discriminate these cells from Langerhans cells. Importantly, when we compared the relative proportions of these cells across different anogenital tissues compared to our abdominal skin, we found they were enriched. So if we look at abdominal tissue, Langerhans cells, as previously reported, are indeed the major mononuclear phagocyte subset in this compartment. But if you compare abdominal skin to genital skin, you can see here that these blue CD11C positive cells are increasing in their proportions. And particularly when we move inside the body to the type 2 mucosa of the vagina, fossa vernicularis, and the anal canal, these cells completely dominate over Langerhans cells, which is obviously a very, very important finding, as these are the actual sites where HIV transmission occurs. So we were very interested in what these cells were. So we carried out detailed RNA sequencing, comparing these CD11C positive epidermal dendritic cells to all known subsets of mononuclear phagocytes derived from tissue. And we found that actually they transcriptionally almost identical to conventional dendritic cell 2s found in the dermis. So we actually hypothesized that these epidermal CD11C positive cells are in fact CDC2s that have migrated into the epidermis, and we're in the process of formally proving that as we speak. Importantly, if I can just direct your attention here to the right-hand side of this graph, the right-hand side most histogram, although we believe these cells are CDC2s, importantly, epidermal CDC2s, shown by the blue graph here, the blue bar, are much, much more potent at antigen presentation than their dermal counterparts shown in red. And this is really important, as it's the antigen presenting cell capacity of these cells, which actually enables them to transmit HIV to CD4 T cells. So indeed, we actually looked at the interactions of these cells with HIV, and we found that compared to Langerhans cells, they expressed much higher levels of the CCR5 HIV entry receptor. Correspondingly, as shown, you can see here the blue bar again, these cells were therefore much more infectable with HIV. Again, correspondingly, again, they transferred the virus to T cells much more efficiently than any other cell type. And importantly, just to show that CCR5 was mediating this process as an entry pathway, we were able to block this effect using the CCR5 blocking drug Maraviroc. And we also developed RNA scope, which meant that we were able to, working with JKSDs, which means we're able to actually visualize these cells, here shown in the inner foreskin, reaching out processes to capture HIV on the surface within 30 minutes of topically applying the HIV to these inner foreskin explants. And again, here's another beautiful picture of one of these cells extending out a process or a dendrite to capture a single particle of HIV on the surface of the inner foreskin. So in summary, we have used real human anogenital tissues to isolate the actual HIV target mononuclear phagocytes from these tissues. And we have shown that these are epidermal CDC2, dermal monocyte-derived dendritic cells, and dermal Langerhans-positive CDC2s seem to be the key HIV-transmitting mononuclear phagocytes in these tissues, and we believe they're key vaccine delivery targets.